Welcome to OsteoBites. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and the Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are talking with Dr. John Ligon from the University of Florida. Dr. Ligon is one of our 2022 Outsmarting Osteosarcoma grant recipients, and he will be discussing his funded work on using RNA nanoparticle vaccines to overcome the immunosuppressive tumor microenvironment of canine osteosarcoma. Thank you so much, Dr. Ligon, for joining us on OsteoBites today. And we are always happy to have Vicki as our panelist. Vicki is an osteo warrior and MIB Agents Junior Advisory Board member. A little bit more about our guest today. Dr. Ligon is an assistant professor in the Department of Pediatrics at the University of Florida College of Medicine. He earned his medical degree from the Baylor College of Medicine in Houston. And after he completed his residency in pediatrics at the University of Texas Southwestern Medical Center in Dallas, he pursued a fellowship in pediatric hematology and oncology at Johns Hopkins University and the National Cancer Institute in Maryland. In the following years, he completed a senior fellowship in pediatric immunotherapy at the National Cancer Institute and another in pediatric sarcoma at Johns Hopkins University. So no one accused you of being undereducated, Dr. Ligon.
Yes, sorry, glutton for punishment.
Yeah, his research interests include immunotherapy, tumors, and bloodstream infections. And before we get started with Dr. Ligon, we have a couple of announcements. In addition to our OsteoBite series, MIB Agents offers many programs for the osteosarcoma community, including Ambassador Agents, which pairs newly diagnosed patients and families with survivor families, Gamer Agents, which gives osteo warriors and siblings a community to connect with and have fun, and Healing Hearts, which offers grief workshops for bereaved families. All our MIB Agents programs are free of charge, thanks to generous donors who make it better with us. Thank you for considering your donation to support these and all MIB Agents programs, and we'll put a link in the chat. And here are some other ways you can support MIB Agents and our programs now and in September for Childhood Cancer Awareness Month. You can join us for our virtual Outrunning Osteosarcoma events. You can run, walk, cycle. You can do it outside. You can do it inside on a treadmill or stationary bike. You can do it with friends, either together or even virtually apart. You can even do parts of your run over weeks or over the course of a month. We have a 5K, 10K, half marathon, and we'll put a reg link in the chat. And then we're also doing our gold bows for the mailboxes again this September. You can make and sell bows, or you can purchase a bow to raise awareness for pediatric cancer and show that you care. And we'll put some more information about that in the chat. And we are very grateful to our sponsor, the Osteosarcoma Institute. This episode is sponsored by OSI, a nonprofit organization led by osteosarcoma experts from the top US cancer centers who together are concentrating on the cure for osteosarcoma. The mission of the OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials in science. And in addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Their osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. This resource is available in English and Spanish and aims to help patients and families find answers to their questions. So thank you so much to the Osteosarcoma Institute, and I'm going to hand it over to Vicki to introduce herself.
Hi, everyone. My name is Vicki Hoy, and as Christina said, I'm a member of the Junior Advisory Board, an osteo warrior, and also a host for the Osteo podcast as well. I was recently very honored that my best friend and friend's father were able to run the Outrunning Osteosarcoma event in my name. So I commend that you all do it as well, even if it's not in my name, but that was recently posted on Instagram in case you saw. Outside of MIB, I'm excited to start at Villanova School of Business in about 3 weeks.
Thanks so much, Vicki. And then Dr. Ligon, if you want to go ahead and share your slides, we can go ahead and get started with your presentation.
Great. Again, thank you guys so much for giving me a chance to share some of the work that we're doing. And again, thank you so much for funding the work that we're doing here at the University of Florida. I am really excited about all the work that we're doing and have done in just the 7 months since I got here. And I think the future is very bright. So today we're going to talk a little bit about how we can use this novel RNA nanoparticle vaccine to overcome the immunosuppressive tumor microenvironment of canine osteosarcoma. I do have one disclosure that I received research funding from Amgen, and there is exactly one slide related to this research in this presentation, but I'm not going to be talking about any off-label uses of drugs or medications in this presentation. And these are our objectives. So to kind of set us up, I want to talk about some of the immunosuppressive mechanisms that cause immunotherapy to not be effective in osteosarcoma. I want to discuss how our novel patient-specific RNA nanoparticle vaccine may overcome these challenges. And I want to provide an overview of the current state of comparative oncology osteosarcoma research at the University of Florida. Now, when I mention comparative oncology research, what I mean is using canine clinical trials as a platform to move some novel therapies forward, because we know that canine osteosarcoma is very similar to human osteosarcoma, and so this is, really is a great opportunity for us to, try new agents in a model that is very similar to human osteosarcoma. So I just want briefly to set up the presentation by talking about this cancer immunity cycle, because I think that the current model of thinking about immunotherapy thinks about kind of having a silver bullet approach of acting at one of these steps and hoping that that will be strong enough to overcome cancer. But while I'm not going to go through each of these different steps, what I'm trying to convey here is there are a lot of steps that go into having your immune system recognize and kill cancer. And sometimes you're fortunate and a single silver bullet, like a checkpoint inhibitor or a CAR T cell, can work. But in other cases, you really kind of need to attack the cancer at multiple steps to unleash the immune system. So I really just want to kind of highlight that this really is a complex process. And the reason why we're talking about this is, as we all know, unfortunately, there's no evidence for single agent immunotherapy being effective in osteosarcoma at this point. This is one slide that shows the data from a recently completed children's oncology group trial looking at an immune checkpoint inhibitor, and there were no responses in osteosarcoma or in any of the other pediatric solid tumors for that matter. So we're really going to need something more than just a single bullet, a single silver bullet, in my opinion, for a lot of these solid tumors. And the reason why is that it's not just the cancer fighting the immune system. You have a lot of other types of cells that are present within your tumor. You have, let me just get a pointer. Great. You have your cancer cells, yes, but then you also have these immunosuppressive myeloid cells like myeloid-derived suppressor cells, tumor-associated macrophages, regulatory T cells, all there that can inhibit your immune cells like T cells and B cells. You have your immune checkpoint molecules that can turn off your T cells and B cells. And you have all these other components that are part of your immune system there as well. And trying to figure out how to overcome all these pro-tumor cells by activating the anti-tumor cells is, I think, the puzzle that we're going to need to solve to overcome osteosarcoma. And another piece that I think is important to recognize is that not all cancers are the same. So different cancers interact with the immune system in different ways. And I like to think of these in three different ways. There are inflamed tumors, which have lots of T cells already present. And these tend to be the ones that respond to drugs like checkpoint inhibitors. There are immune excluded tumors where all of your T cells are kind of at the periphery of the tumor. And then you have immune deserts where there really are no immune cells present. And for the longest time, people have really thought of osteosarcoma as being immune deserts because a lot of the earliest papers looking at osteosarcoma said that there really was no immune activity present. I'm going to try and argue that osteosarcoma, and particularly the pulmonary metastases, which, as we all know, are how our patients ultimately die from osteosarcoma, that they really look more like an immune-excluded tumor microenvironment. And so I think we need to think of osteosarcoma as this kind of tumor if we're going to design immunotherapy that's going to be effective. So all of this really started from a observation that we made at Johns Hopkins, where just counting up the number of immune cells that are present in our sarcoma samples. We have a large biobank of sarcomas at Johns Hopkins that we've been studying over the years, and a simple observation showed us that osteosarcoma, while the primary bone tumor does not have very many of these immune cells in the tumor, if you look at the lung metastases here, you actually do see much higher infiltration in the metastatic tumors compared to the primary bone tumors. In fact, in some ways higher than any of these other sarcomas that I have on the screen. So this really led to our initial hypothesis that the tumor microenvironment of these pulmonary metastases must be very different from the primary bone tumor, and that the tumor microenvironment of the pulmonary metastases must be highly immunosuppressive and prevent immunotherapy if you have all of these immune cells that are present and you're not getting an effective response. So to test our hypothesis, we used 3 different ways to look at the osteosarcoma tumor microenvironment. And I'm going to go through some of this data, and I'm going to do it relatively quickly because I do want to get to talking about the RNA nanoparticle. And ultimately, what I want to convey is I think that some of the preliminary data that we've generated shows why the RNA nanoparticle really may specifically overcome the challenges that are present in the osteosarcoma tumor microenvironment. So the first way that we look at the tumor microenvironment of osteosarcoma is through immunohistochemistry, which is simply putting the tumor on a slide, staining it for different markers, and seeing what it looks like under the microscope. Initially, we did exactly what everyone else has done, and we looked at the primary bone tumors, and indeed, we found that these look like immune deserts. So CD3 is a marker of T cells, PD-L1 is an immune checkpoint molecule. You don't see any of these markers present in the primary bone tumor. Really, the only marker we saw in any appreciable amounts in the primary tumors was CD163. And this is a marker of myeloid cells, and usually the bad kind. So these are the kinds of myeloid cells like the myeloid-derived suppressor cells, tumor-associated macrophages that I talked about a few slides ago that can be immunosuppressive. So this was not surprising. This was similar to what other groups had seen before. However, when we looked at the pulmonary metastases, we actually saw something that was quite different. So if you look at the tumor interior, yes, it looks like a immune desert, kind of like the primary tumors. But here at the interface region where the tumor meets the normal lung, if you zoom in on that, you have a bunch of these T cells present. Here I'm looking at CD8, which is another marker of lymphocytes, and specifically the cytotoxic T cells, or the ones that can kill cancer. And you have a lot of them there. They're just all stuck at the edge of the tumor. And if you put up a corresponding slide of PD-L1, that immune checkpoint molecule, you actually see that it's kind of a mirror image. It's as if the tumor recognizes that these T cells are there, they're trying to come in and kill the cancer, and so they're putting up this focal PD-L1 shield to stop the immune attack. That's all a very nice story. It makes perfect sense. But of course, we know that simply treating with an immune checkpoint inhibitor hasn't cured osteosarcoma yet. So to really get down to understand the tumor microenvironment, we expanded our studies and looked at a number of other markers. The first thing that we did was we quantified a number of markers of these T cells. So CD3 and CD8 are both markers for T cells. And when we quantified this, what we showed was that indeed the pulmonary metastases in blue had higher infiltration of both kinds of T cells compared to the primary bone tumors. And then if you focus specifically at that tumor normal lung interface and kind of zoom in on that area, the concentration of the T cells goes up even more. This is also true actually for FOXP3, which is a marker of regulatory T cells and is a kind of immunosuppressive T cell. But if you look at the numbers along the side, there's a low number here. I don't think that the T regulatory cells are really what are inhibiting the cytotoxic T cells, but I do think it's an interesting point and something to point out. Next, kind of looking at those myeloid cells that we saw also abundant in the primary tumors, we see that indeed they are also in the pulmonary metastases, and kind of like the primary bone tumors, they're present throughout the entirety of the tumor. So I'm showing you CD163, which is the marker that I showed you previously. CSF1R is another marker of these myeloid cells. There actually are some agents entering clinical trials targeting CSF1R as well. So that's why it's a particularly interesting marker to look at. And if you quantify these numbers, there really is no difference between the primary bone tumors, again in red, versus the pulmonary metastases in blue. They were maybe slightly more concentrated at the interface, but really the story here is that these myeloid cells, which are immunosuppressive, are really present throughout these tumors. They're in primary tumors, they're in the metastases, and you can find them throughout the entirety of the tumor. Finally, we did look at some of those immune checkpoint molecules, and in addition to PD-L1, which we showed previously, we looked at some other markers. And again, I think it's important to know that there are drugs that target each of these immune checkpoint molecules that have entered clinical trials as well for adult cancers, TIM3, LAG3, and IDO1. And here, it really does look like these kind of all line up here at the interface to varying degrees. And if you quantify them, this was true for all of these markers, that they were all significantly more present at that interface region compared to the interior of these pulmonary metastases. So to kind of summarize the immunohistochemistry, we see that there are T cells there, but our next question, and we see that they're kind of stuck at the edge of the tumors. The next question is, are these T cells active? Could they be capable of killing cancer cells if they weren't being stopped by the tumor microenvironment? To answer this question, we did multi-parameter flow cytometry looking at the markers on the T cells that we isolated from these tumors. When we did this, we first of all confirmed what we saw by immunohistochemistry, that indeed these cells all seem to have higher expression of these immune checkpoint molecules. Just to briefly orient you, for all of these slides, again, the primary bone tumors will be in red, the metastases or the lung tumors are going to be in blue. And so for CD8 T cells, you see that there's higher PD-1 expression. There's also higher expression of this other immune checkpoint molecule called LAG3. And this is important because the literature shows that the T cells that express these immune checkpoint molecules are also likely the ones which are capable of killing cancer if they were not being restrained. And indeed, as you look at some of the other markers of activation, like interferon gamma, again, in your CD8 T cells, you have higher expression. So this really shows us that, yes, you have T cells. Yes, they could kill cancer if they weren't being restrained. So what is keeping these T cells from working? And so to answer this, what we did was we did something called differential gene expression profiling. And let me explain a little bit in greater detail what that means. So, again, we've been talking about this interface region where all of the T cells are kind of getting stuck at that tumor normal lung interface. So what we did was we took a piece of the tumor, put it on a special slide called a pen membrane, and we used a laser to dissect out these two regions, the interface region against the interior. Then we isolated all the RNA from these two different sections and then performed a study called RNA-seq to look at the genes that are expressed in the RNA to give us an idea of what kinds of programs are being run in this interface region that are preventing the T cells from getting further in. So at first, as you can imagine, trying to decipher what all the data from like 30,000 genes is difficult, but we have some tools to help us understand it. And one tool that we use is called CyberSort. So this helps us to kind of get at a bird's eye view, what are the immune cell populations present? And you have all different kinds of immune cells along the top here. You have B cells, you have T cells, you have several different kinds of myeloid cells. And again, just to orient you, here, blue is going to be the pulmonary metastasis interior, and green is going to be the pulmonary metastasis interface. Again, the interface being that important region right where the tumor meets the normal lung, where the T cells are being stuck at the edge. And at first glance, indeed, it looks like we confirm again what we were seeing through immunohistochemistry, that we have higher expression of genes related to T cells here at the interface compared to the tumor interior. And then if you use another way to kind of decipher the genes that are being present called XCell, which looks at specific genes and can give you an idea of what types of cells are expressing those genes, you see that a number of these genes which are related to cytotoxicity or being activated, which means that they could kill cancer, these are all preferentially being expressed in the T cells, as well as these immune checkpoint molecules, which again are important because that shows us that these T cells that express these checkpoint molecules are usually the ones that are able to kill cancer if they're not being kind of restrained by the tumor microenvironment. So all these different genes, which all tell the same story, that these T cells really have signs that they could kill cancer if they weren't being held at bay. Additionally, and I'm just kind of slipping this in here because that's going to be important when we talk about the RNA nanoparticles, we also saw that a subset of genes related to dendritic cells were also present at the interface region compared to the interior. And these are very, very, very important because dendritic cells are what are called antigen-presenting cells. In order to for the T cells to become active, they need these antigen-presenting cells to kind of show the cancer proteins to the T cells. And so you really need both of them there in order to have a productive immune response. So something is keeping both the T cells and the dendritic cells kind of stuck there at the interface region. So going back to CyberSort, I've been focusing on the T cells up to this point, but even louder than the T cells is this picture that these M2 macrophages are just dominating the entire tumor microenvironment. The interior, the interface, and M2 is generally another way of saying tumor-associated macrophage, myeloid-derived suppressor cell, bad, pro-cancer myeloid cell that is going to turn off your T cells. So, and as we dug into them a little bit more deeply, I'm going to go through this a little bit quickly, but we really found that genes related to the granulocytic form of myeloid-derived suppressor cells seemed to be the ones that were present at the interface. So these might be kind of the myeloid cells at the front line, so to speak, that are keeping the T cells at bay. Finally, and to kind of get to what this means for our patients, we showed that the interface was very important for patient outcomes. So all of what I'm showing here are data just from the interface region. And we found that if you looked at our immunohistochemistry data, patients with higher expression of these different immune checkpoint molecules tended to have worse progression-free survival. And then same for patients who have gene sets that are kind of indicative of productive immune response, gene sets related to CD8 T cells, T cell survival, MHC class 1, which is an antigen presenting cell signal. These patients all actually did better. So this really shows, again, that the immune system is important for how patients do in fighting cancer. So in conclusion for this first part, we showed that osteosarcoma pulmonary metastases are an immune excluded tumor microenvironment compared to the immune desert of primary bone tumors. And this seems to be related to these M2 macrophages. So we're going to need to find a way to reprogram these immunosuppressive myeloid cells in order to really unleash our T cells and kill the cancer. So this is where I think our RNA nanoparticle could come in. So thanks for bearing with me for a little bit of that exposition, but I think it's important because the RNA nanoparticle, I think, really kind of tackles some of these issues head on.
Yeah, that was a really good setup. Thank you.
Perfect. To understand a little bit of exactly what this RNA nanoparticle vaccine is, you actually take a patient's tumor, in this case, osteosarcoma pulmonary metastasis, you isolate all of the RNA from the tumor, and then you attach it to this lipid and a nanoparticle carrier. You make more of them, and then you actually give it intravenously to a patient with the idea that it's going to reprogram a cold tumor into a hot tumor and activate your immune system throughout the entire body. And what it really does is that it works through those dendritic cells that I was talking about earlier to activate your T cells and enable them to finally have the anti-tumor activity. And I think what's really important to recognize about this approach is that we have what's called a multilamellar nanoparticle. And I think right now when people think of mRNA vaccines, they think of the COVID vaccine. And in some ways we're similar, but in other ways we're quite different. And when I say multilamellar, we're not just kind of expressing RNA in one layer. We have kind of multiple alternating layers of positive charges, then RNA, then another layer of positive charge, then another layer of RNA. And why is this important? Well, the problem is that the whole reason that cancer even exists in our body is that it's found a way to trick the immune system into thinking that it's not dangerous. Because otherwise the immune system would have recognized it as dangerous long ago and wiped it out. Here, by kind of having this shell that looks like an onion in some ways, if you look at it under a microscope, it actually looks very similar to a virus. If you looked at a virus under a microscope, that's what this would kind of look like. And so it transforms the cancer and the RNA that it expresses into something that looks much more dangerous. So now all of a sudden the idea is that your body is going to see this and say, hey, this does look like something that's dangerous. We need to pay attention to this. And to show a few of the experiments that we've done using these nanoparticles, we actually can have shown that the RNA nanoparticles do seem to reprogram the tumor microenvironment. When you give these tumor-derived nanoparticles to mice which have a model of osteosarcoma, you wind up seeing depletion of these tumor-associated macrophages and myeloid-derived suppressor cells. And importantly, you also wind up having significant activation of both your dendritic cells and your T cells. So it's kind of working on both axes, which is really important. Additionally, what we showed is that it winds up having an impact on these mice in terms of survival. So whereas most mice which are injected with osteosarcoma wind up dying, if you give the tumor-associated RNA nanoparticle here in blue, actually you wind up curing most of them. And then at the end of your experiment, if you look at their lungs, just kind of after the to look at the effect on the lungs, the ones who were treated with RNA nanoparticles, their lungs are completely clear, whereas the untreated lungs are completely riddled with osteosarcoma. So this is really a pretty remarkable outcome for the mice in this model. Importantly, what we showed actually in a different model, this is a brain tumor model, is that in this case, the RNA nanoparticles go to the myeloid cells that are close to the blood vessels. And it's a little hard to look at this. I don't know why this wound up being a little bit off, but basically this red marker is the RNA nanoparticle. The green is a marker of the vasculature. And we know that the myeloid cells tend to hang out right by the vasculature. And so if you superimpose them, your red and your green are right on top of each other. So this really shows us that we think that the RNA nanoparticles are going to exactly where the myeloid cells are, and actually activating them to become functional dendritic cells, which is what I'm showing here on the right side of the screen. This CD86 is a marker of dendritic cell activation. Additionally, you see upregulation of these genes that are all important for having effector dendritic cells, and in fact, for reprogramming the myeloid cells. And just going through this a little bit quickly, we think that this is related to a gene that's known as interferon alpha. And if you use specific kind of antibodies that block interferon alpha, you show that you kind of lose all of the activity of the RNA nanoparticles. So just to give a little bit of information on the mechanism that the RNA nanoparticles use. And I'm going to skip that as well, because I want to talk a little bit about our comparative oncology canine model. So here at the University of Florida, we have a collaboration between our College of Medicine and the College of Veterinary Medicine, where we've been able to treat canines with spontaneous or kind of osteosarcoma that happens in real pet dogs, and use our RNA nanoparticle vaccine to target both brain and bone tumors. And this is Rowan Milner, our very good collaborator, who's the head of these protocols. And what we have found is that in brain tumors, we really have seen a remarkable extension in the survival for the canines who've received the RNA nanoparticle. So historically, canines with gliomas all die within 50 days. In our patients who've been treated, there's actually a very significant extension in their survival, which has not been observed in canines that don't receive this treatment. So this really is remarkable, and this is also important for osteosarcoma because we know that brain tumors also have the same problem of immunosuppressive myeloid cells. So this led to our hypothesis that RNA nanoparticles, our hypothesis that we're going to be testing with the Outsmarting Osteosarcoma grant, that RNA nanoparticles will reprogram the immunosuppressive myeloid compartment in recurrent osteosarcoma, and that the administration will be safe and effective for improving event-free survival. So in aim 1 of our grant, which we will be testing, is looking at the safety and the efficacy of this RNA nanoparticle vaccine. And we're particularly interested in this combination with radiation. So safety and efficacy, because we want to, I should have mentioned that some of the data that we've generated up to this point has led to the RNA nanoparticles entering phase 1 trials for adults with brain cancer. So we want to expand treatment with this drug to patients with osteosarcoma. And so if we can get safety and efficacy data for canines with osteosarcoma, I think it will help us to move this drug into clinical trials for patients with osteosarcoma.
So exciting. And so for safety and efficacy, though, was established for the dogs with brain tumors, right? Already going to human trial?
Correct. Yeah. And I think we are So yes, they were safe and had signs of effectiveness in dogs with brain tumors. I think that we need some data for canines with osteosarcoma, specifically just because you have a different site where the cancer is, and so you want to make sure that you don't have unexpected toxicities when you're treating cancer in a different location.
And were there, in terms of safety, what type of side effects are there? Are there any?
Yeah, so that's a really, really good question. So at any time that you use immunotherapy, you kind of run the risk of kind of supercharging the immune system. And I think that you see that with things like CAR T cells, where you have toxicities like cytokine release syndrome. And I think I actually have it on the next slide even. I'll come back to that. So that within just a few hours, we actually do wind up seeing in canines significant immune activation. So this is within 2 or 6 hours. You see these dogs start running fevers. You start having significant activation of both dendritic cells and T cells in the blood, which you kind of wind up seeing a little bit of this cytokine storm. It really is pretty remarkable. Particularly in dogs that we've tried treating in combination with chemotherapy, you can wind up seeing significantly decreased blood counts. So, just like with chemotherapy, for example, you have low blood counts. This seems to be exacerbated when you try to combine it with RNA nanoparticles. So canines had really, really low platelet counts. And so for this reason, we actually have decided not to combine it with chemotherapy in our canine osteosarcoma trial. Yes, you do wind up having some of these kind of inflammatory reactions. At least thus far, the inflammatory reactions like fevers have resolved relatively quickly. The blood counts can remain low for a while, especially if you did combine it with chemotherapy. So this is going back here just briefly, because I know that I've been talking too much. This is kind of an exceptional case where we had a patient, a canine patient who had a brain tumor, who got the RNA nanoparticle vaccine and then was treated with radiation. And so you can see this was the original tumor here on the right, and this is the tumor after they received this combination. And you can see that there's a significant reduction in the size of this tumor compared to this one. And from speaking with all of our veterinary colleagues, they say this does not happen with radiation all by itself. So there is some significant interest in thinking, just like other kinds of immunotherapy, maybe this RNA nanoparticle could be a way to sensitize osteosarcoma to radiation treatment. And so this is a particular combination that we're interested in exploring in our canines with osteosarcoma. And from speaking with some of the radiation oncologists and veterinarians over at the veterinary school, it sounds like a number of owners choose to pursue radiation therapy rather than amputation for their dog's osteosarcoma. So I think that this will be a particularly interesting combination that we can look at in these canines. And then this second aim really is to look at the immune changes in the tumor microenvironment. So these are all just markers that we would measure from the blood, or vital signs in this case, to look at signs of inflammation. But in addition, we would also be interested in collecting biopsies both before and after treatment with the RNA nanoparticle. And then, kind of similar to how I was showing you at the beginning of the presentation, trying to see how does the tumor microenvironment change from the pre-treatment to post-treatment. Do you see this macrophage signature going away? Do you see the T cells starting to invade the tumor interior? And so these are some of our radiation oncology colleagues that I have highlighted here on the left. These are both veterinarians who take care of canines with osteosarcoma. And a really interesting fact that's important is that dogs who get radiation treatment have to undergo anesthesia to get radiation therapy to make sure that they don't move. And so this provides us a great opportunity to get another biopsy from these tumors in a way that we wouldn't necessarily do for humans. So this is a great way to look at how the tumor microenvironment affects things.
You might be getting to this already, Dr. Ligon, but how is the radiation and the RNA nanoparticles sequenced? And then are you able to then get samples? I mean, I know we don't want to, it's not easy to get biopsy samples, but is there a way where you can get samples where it's, depending on how it's sequenced, where you can see effect after radiation and then see how it changes once you add the RNA nanoparticle?
That is an excellent question, and it's actually something that we're talking about with our colleagues. I think it's a debate that we're having internally, because the dog that we have kind of observed previously got RNA nanoparticle and then radiation. There is some other literature that shows that if you sequence things with immunotherapy and radiation, that it's better to do radiation first, because if you give immunotherapy first, you're going to theoretically activate the T cells in the tumor, but then when you give radiation, you're just going to kill all of the T cells that you just activated. So I think kind of our experience makes us think about doing RNA nanoparticles first. At the same time, mechanistically, it kind of makes sense to do radiation first. So we are still trying to figure out what makes the most sense. Additionally, and this is something to consider for the future, in addition to kind of like this overarching picture of the genes that are activated, what we would really love to do is see what's happening in the tumor microenvironment at a granular level, and see how all of the immune cells are interacting with each other. And it would also be great if we could determine who is or is not going to respond to treatment. So a lot of people are looking at different ways to kind of model cancer in a three-dimensional model. And so in general, what you do is you kind of take a tumor, take a area of interest, and kind of embed it, and then put it on a slide. Or you embed it, you give a treatment, and then you kind of look at it on a slide after the treatment to see how your treatment affected things. And you can look at a number of different ways of analyzing the response to treatment, either just looking at, did the cancer die? Do you see markers of activation? Do you see changes in your gene expression profiling? And one model that we are, or one of my collaborators here has a really outstanding model for cancer. His name is Greg Sawyer, and he has what's called a liquid-like solid tumoroid model. So what he does is you take a tumor, mince it up into very, very small pieces, and then you can kind of inject a dye into the tumors, or various dyes to mark different parts of your kind of mini tumor. So in this particular example where we were looking at glioblastoma, you have the tumor in blue, the RNA nanoparticle in red, and you wind up seeing the RNA nanoparticle kind of having its effect here with green through a mechanism that's a little difficult to explain. But essentially, you're able to see kind of in real time on a individual cell level these changes taking effect. Additionally, here you can see the tumor in green, and then your immune cells here in red. And over a time lapse, so 0 hours, 4 hours, 8 hours, you see the tumor starting to die. So you have a lot less green here at the end compared to the beginning. And what we think is really exciting is that you can get a video of this in real time. So here I'm showing you some of the experiments that we've been doing in AML with my collaborator, Zhangping Huang, and then Dewey Win, who works with Dr. Sawyer. I'm just going to briefly show you guys this video because I think it's really cool. And I would love to make a similar video like this in osteosarcoma. So this is not osteosarcoma, this is AML, which is a type of blood cancer. But you can see here, we have the AML in red, and then you have a CAR T cell here in green. And so just for example, take a look at this red dot there. You can see all of these green CAR T cells kind of swarming around it. And as I press play, you can see that they wind up making the AML go away by the end of the time lapse. Now another one comes a little bit later, but or actually no, it just skips back to the beginning, so it's back. But if you kind of look here at the very end, that AML cell has been killed. So I would love to do this with osteosarcoma, and we actually recently received additional funding from the Hyundai Hope on Wheels Foundation to try and do this in osteosarcoma. So this actually is a tumoroid of a human osteosarcoma lung metastasis. So here, just in very simple terms, green is alive, red is dead. So you see that most of this is alive. And you see that we were able to maintain both pieces of the lung as well as the tumor and that tumor normal lung interface that I was talking about at the very beginning of this presentation. So I really think this is a great model so that we could potentially then make RNA nanoparticles from the same patient, treat these tumoroids with this model, and see how it affects the immune activation of these cells. And all this work was done by Sadim Qudasat, who is a PhD student here at the University of Florida. I think this is a really exciting opportunity. And what we would love to do, actually, I don't know if yes, all right. So I showed you here a human tumor, but I would love to do this with our canine tumors as well for the canines who are on the clinical trial. Because what I could do then is actually, when we enroll the dog on the trial, I can take a piece of the tumor, go make a tumoroid, and then do an experiment in the next 2 or 3 days and find out, does the tumoroid respond to the RNA nanoparticles? And if it does that predict that the dog will also actually respond to the RNA nanoparticle? I think that would be incredibly helpful to determine whether or not this treatment would be effective eventually once it makes its way into humans as well.
Couple of things that's like so fascinating, and big advantages here I think are the time advantages, right? But I'm curious, like, how long does it take to create the vaccine once you have the tumor tissue?
Mm-hmm.
And then it sounds like the canine tumor development is also really fast. But can you quantify that a little bit?
Sure, absolutely. So to answer your first question, for canines, the vaccine development really just takes a couple of days, something between like 3 and 4 days. Usually we could treat as early as like a week or 2 after the surgery. The tumoroid, there are a couple of advantages to this as well. So one, you could make it kind of directly after the surgery. So this tumor, this picture came from the same day that the patient had surgery. So we were able to establish that on the same day. But if for whatever reason you want to save it and do the experiment later down the line, you actually can freeze these tumoroids down and then establish it in culture later when you're ready to run the experiment. So say, for example, if you needed to wait a few days while you're producing the vaccine, that still works. in humans for our human clinical trial, thus far it's taken, I believe, somewhere between 5 and 8 weeks for production of the RNA nanoparticle vaccine. Just because it takes a little bit longer time to kind of get things through all of the checks to make sure that it's safe to administer to patients. So kind of returning, and this talks about what I was just saying, that these tumoroids give us a chance to establish tumoroids from canines enrolled in our trial, image the RNA nanoparticle interacting with the canine tumor in 3 dimensions, and then hopefully identify canines with the best opportunity to respond based on our model. And then, of course, our ultimate goal is to bring this new treatment into human patients with osteosarcoma. And this is our planned human clinical trial, which we actually are, I believe, very close to getting funding to do some of to move the human clinical trial forward. So hopefully we will be able to open that through the National Pediatric Cancer Foundation sometime in the near future and really make this a treatment option for human patients. So in conclusion, I think in terms of the future, I hope I convinced you that these immunosuppressive myeloid cells are a major obstacle and we need to meet them head on in order to design effective immunotherapy. I think these RNA nanoparticle vaccines may allow us to reprogram those myeloid cells. And I think really importantly, these comparative oncology clinical trials supported by MIB agents will be key to advancing this treatment into clinical trials. And then I didn't really talk about it here, but I think these tumoroids are also a great opportunity to study rational immunotherapy combinations, and see how else we can kind of move this novel treatment forward in combination with other treatments. So with that, I have so many people to thank, both from my training at Johns Hopkins and the National Cancer Institute, and all of my collaborators here at the University of Florida. I would specifically highlight Elias Sayor, who's my mentor, both in general and in life, and also specifically on this grant, as well as Dwayne Mitchell, and then all of the other collaborators that I've kind of called out during this presentation and that you see on this slide. And again, thank you so much, Christina, for funding our work. And I really think this is going to, I'm really excited about this treatment and about what we can learn through the research that you guys have funded. So thank you so much.
Thank you. This looks, it's so interesting, and it's so encouraging that it's already in canine trials. And also encouraging that you have the data from, I know, a different type of tumor, but still, that's gives great hope for osteo.
Absolutely, absolutely. So they actually just treated the second human patient with brain tumors within the last couple of weeks. So slowly but surely, getting the kind of safety data that will hopefully let us move this into patients with osteosarcoma.
I just had a question. How long does the reprogramming last, and how often do you have to give this booster vaccine once you've made it for the patient?
Yeah, so that's a really great question. And the honest, answer is that we don't know. Right now, the way that we have the trial designed is that we would initially give the treatment weekly for the first 2 doses, and then it would be every 2 weeks for the vaccines 3 and 4, and then kind of continue it monthly for about a year to kind of keep giving booster doses to try and keep reprogramming the myeloid cells to be good antigen presenting cells. And the idea is that over the course of the year, hopefully you will either kill the cancer or at least get your T cells activated to the point that it can hold the cancer in check. But we really don't know. So I think that getting some of the post-treatment biopsy samples will be really important for answering that question.
I'm wondering. Oh, sorry, Vicki, go ahead.
I was just going to ask another question. Is this possible to be used on kind of initial cases of osteo that have already metastasized to the lungs, or is it not good to use since it's going to be in the a limb as well?
That is also a really, really good question. And it's something, honestly, that we internally debated a little bit. Should we be ambitious enough to, try proposing this in patients with metastatic osteosarcoma when they first present? And because, we know that those patients also are in need of something additional to treat their tumor. What we ultimately came down on is that this is such a novel treatment, that has literally been used in 2 human patients so far, that for a phase 1 trial, we really felt that likely the bar is going to be a little bit high to include newly diagnosed patients, and I think that it would be kind of more reasonable to agencies like the FDA to propose a trial in patients who have relapsed disease. But I think if we have a strong signal here in these initial studies, it's absolutely something that we could consider in patients who are newly diagnosed.
And I'm curious, Dr. Ligon, I wonder, so I mean, ideally what you're going to see is that there's no progression, right? That you keep giving the boosters and you don't see progression. But let's say you do see a relapse or progression. Like, is it a possibility to create a new vaccine based on the new tumor and try that again?
That's a really interesting question. Theoretically, no. Well, I mean, so theoretically, yes. But like realistically, right now they would come off of the study. But I'm trying to think now about whether we could, offer that as a potential, second treatment. And I'm gonna have to think about that a little bit more, because that's a really interesting idea. Now, yeah, because what you are, bringing up is, very relevant. That when tumors progress, it means that they've changed, and ideally, you would be giving a vaccine that kind of represents exactly what the tumor is right now. So if I made a new vaccine with all the changes that the cancer has kind of incorporated, maybe you would wind up getting a response to the new formulation. It's a really interesting question. I can't come up with an answer in the last couple of minutes here, but I'm going to think about that a little more.
I mean, just because of the timing that you mentioned too, with being able to produce the vaccine relatively quickly and even test it on the new tumorides, it might be, because obviously that's always but if you could address it in the same way, then it doesn't matter if it keeps coming back. If you can just keep, it's like the go for
whack-a-mole,
game
yeah.
at the arcade
Yeah.
where you just pop it again with the hammer. So yeah.
That's really interesting. We should definitely we should try that in some of our models. That's a great idea.
Well, thank you so much, Dr. Ligon. This was so interesting and helpful. And I really appreciated also all the slides at the beginning that really helped explain the tumor microenvironment in a way that was kind of really easy to understand. So thank you for that. And thank you for making it better for pediatric oncology patients everywhere. You can find more information on this and all OsteoBites on YouTube, on our website at mibagents.org, and at your favorite podcast place. And next Thursday, please join us on OsteoBites. We're going to be talking with Dr. Jason Eustein, a professor at Emory University. And Dr. Eustein is also one of our 2022 Outsmarting Osteosarcoma grant recipients. And he will discuss the work that is funded by this grant, combination therapies to improve immune-mediated approaches for osteosarcoma. And if you have any ideas for future OsteoBite episodes, please share with us. I put an email address in the chat. Please send us any ideas. Thank you again, Dr. Ligon and Vicki, for spending an hour with us today, and to our sponsor, the Osteosarcoma Institute. And thank you all for joining us on OsteoBites, and we hope to see you next week when we chat with Dr. Eustein. And in the meantime, please go to our website at mibagents.org and sign up to outrun osteosarcoma through September. Thanks so much, everyone.